Are we ready for liftoff?
Yeah.
Excellent. Okay, well, I just want to begin by thanking Uta and Tanina very much for this wonderful opportunity. I look forward to this workshop every year because the enthusiasm of the students and the very thoughtful way that the curriculum is put together, I think is quite exceptional, and it's such a privilege to be part of this very unique instructional opportunity. Without further ado, I wanted to give everyone sort of a layout of the things we'll be discussing today. I'm going to give, since I think, the first speaker of the entire workshop, I'll give a brief primer on EVs. I think everyone has a general idea of what EVs are, but it always helps to visit the basics. Then I'll talk about why EV therapeutics are such a promising platform, um, despite the fact that we've faced some challenges. Then I'll talk about sort of the four venues by which we as researchers are trying to translate EVs to the clinic. And then I won't steal the thunder from my remaining colleagues, and I'll talk about instead a novel drug discovery paradigm that my collaborators and I have been working on for the past, I want to say 15 years, in mining EVs for RNAs and letting them sort of be our teachers as to what RNA molecules are important for therapeutic application. Then I'm going to zoom back out and talk about the broader challenges facing all the different applications of EVs to the clinic. And then I'll look ahead with some perspective ideas. This is not an exhaustive presentation about the challenges and the opportunities that lay before EV therapy, but I hope that this will be a nice opener to what would be a very long and very fruitful discussion. So EVs, in very general terms, are these lipid nanoparticles that come from two different classes. They're either shed from the membrane, and that includes dead and dying cells, cancer cells. We call them ectosomes because they're just passively shed. And then the other ones are the exosomes, which are, they arise from the late endosome, and they're very specifically sort of packaged as part of this very deliberate pathway. And one of the biggest challenges facing EV basic investigation is how to separate these two. And this heterogeneity you'll hear about throughout this course. There's also this issue of lipoproteins. Are they EVs? So to some degree, there's the discussion for what constitutes an EV remains very much open. But in very general terms, EVs are either ectosomes shed from the plasma membrane, or they are deliberately packaged through the late endosome, the escort pathway, and those are called exosomes. So I want to talk about the role of EVs in biology, and I think that's very important because once you grasp the incredible scope and the niche that EVs play in all levels of biology, it becomes easy to appreciate why there would be such amazing therapeutic platforms. So the first sort of role is to instruct. And those we understand from the fields of regenerative medicine, from cancer, from development, and they have cargo that can affect the proteomic, transcriptomic, and even chemokinetic behavior of the recipient cells. They also provide nutrient sources. Bacteria that live in aquatic environments in the ocean very often will secrete EVs that either sequester nutrients from the environment or secrete themselves nutrients to feed nearby bacteria. So that's one other role of EVs in biology. And the third one that I'd like to focus on is protection. There have been many papers talking about the transfer of viable mitochondria in EVs from, say, macrophages to injured tissue. They're also used to eject toxic waste products. It's been shown that some tumor lines will eject microRNAs that limit their growth. They also act as mediators of resistance. So some tumors will secrete products that enhance the resistance of nearby tumor cells. in the face of chemotherapeutic exposure, and then thwarting external danger. So, for instance, some bacteria will secrete EVs as sequesters of antibody molecules during infection. And so one can immediately appreciate the multiple layers by which EVs function in biology. And what I hope to impress upon everyone today is that the role of EVs in therapy is no less diverse than their roles in biology. As you can tell, they're very evolutionarily conserved. And so, if we were to understand how EVs work in therapy, we need to appreciate the multiple functions that they have in nature, because more than likely that nuance transfers to each of the EV populations that we work with. Just to drive the point home a little bit, EVs communicate broadly across all levels of physiology as well, including development, activation of immunity, injury and healing, infection, cancer, regeneration or degeneration, as well as aging and senescence. They're also mediators of communication between different individuals, as intimate as maternal fetal, commensal and host, even in nature, plant pollinator, pheromones, the rhizosphere, which is this symbiotic relationship that exists between root systems, bacteria, and fungus. They also play integral roles in parasite-host relationships, predator and prey, and virus and tissue. And because of their presence in nature, in the biotic compartment, which is inseparable from the abiotic compartment. So you'll find EVs in soil, in oceans, in bodily fluids. You'll find them in nectar, in honey, in milk, in beer and wine, and every single every single medium where that interfaces with a biological entity. So we're surrounded by this cacophony of EVs and this silent orchestra that really drives the point home of just how important these medium of communication are. Okay, now from a conceptual framework, I like to see EVs as the language of every single cell. And the reason why this linguistic framework is helpful is that it helps us understand the inherent nuance and diversity of EVs. What you're seeing here are the RNA distributions of two different EV populations from two different cell types. These are cardiosphere-derived cells. These are therapeutic cells that I'll expand on a little bit later. And those are control therapeutically inert EVs from fibroblasts. You immediately appreciate that the distribution of the various RNA classes are very different. So fibroblasts are chock full of, I can't see from of tRNAs, while they're more sort of evenly distributed among the classes in CDC EVs. And so even though EVs share structural and functional similarities, their language is different. And I like to compare it with the different languages that we speak. Yes, there are similarities. There are some sounds that are different, but very often the meanings are different, the syllables are different, and I include just different delineators between EVs, including tissue-specific, cell-type-specific, stimulus-specific. So a stressed cardiomyocyte will secrete EVs that are very different than a healthy cardiomyocyte. Disease-specific, so a cancerous fibroblast will release EVs that are very much different than a non-transformed fibroblast, age-specific, and so on and so forth. So why EV therapeutics? Well, the rationale from that derives from our initial interest in regenerative medicine. Regenerative medicine is by its very nature a complex biologic, meaning that it's not one inhibitor for one target, but a very concerted set of signals with what we call signaling coherence, meaning that they have different roles to play, but the overall phenotype is the same, whether it's healing or metastasis or whatnot. And this is depicted by this mosaic that you see here. But there are also some advantages that lend them to therapeutic translation, including the fact that they're abiotic, so they're unchanging. So they're not like cells that can change under different conditions and are rather finicky. They're also very structurally stable. They're allogeneic, meaning that we can have one human product for multiple patients. So there's no concern about rejection. In fact, many of the data that you'll see later are actually xenogeneics. So these are human EVs being applied to mouse, rat, and pig, and they function just the same. They also offer a flexible therapeutic capacity. So you can only inject so many cells without running the risk of an embolism, for instance. But with EVs, because of their size, your ability to inject more EVs is a lot wider. And finally, the safety profile is optimal compared to not only cells, but also some synthetic products where some lipid transfection reagents have been shown to be nephrotoxic and toxic to the liver and so on. They're also a modular platform, meaning that you can switch and add different features to those EVs. And the one thing that I want to focus on, at least for the work that we're doing, is that they're deconstructable, meaning that we can pare them down, tear them down to the struts, find out what cargo are functional, and they themselves can be standalone therapeutics. So they're very, very versatile biologic material. And today you'll be hearing about the application of native therapeutic EVs. You'll be hearing about engineered EVs, and that engineering spans the gamut from changing the membrane receptor to the cargo to whatever else. And you'll also hear about EVs not being used as a therapeutic per se, but as a carrier for drug delivery. And those are the three amazing speakers that will be speaking on that topic. So I won't expound too much on that. In fact, I'll focus on a third, or rather a fourth, application of EVs, which is EV-based sort of therapeutics. So we begin with a therapeutic cell. We take the EVs, we mine the cargo for novel RNAs, and then based on their bioactivity, we can begin to create our own synthetic RNA therapeutics. These are not RNAs that we isolate from the EVs, but rather we identify the sequence and then we make them synthetically using solid state chemistry. So why RNAs? Well, because EVs are full of them, and they're full of bioactive RNAs, many of which we know very little about. So to go back to that linguistic framework of EVs, the idea is to decode what these RNA signals are trying to do. And very often, because we know very little about them, what they offer to teach us is not just a new therapeutic, but also a new mechanism of biology, a new mechanism of transcriptomic influence. And this becomes even more pronounced when you realize that 98% of the genome is non-coding RNA. Only 2% codes for genes. The other 98% is preoccupied with more or less regulating that 2%. So that's unprecedented reach. When you think about, and less than more than 90% of these RNAs are unknown. We don't know how they function. We understand microRNAs, siRNAs, long non-coding RNAs maybe, but the remainder of them are by and large obscure. And when you think about, drug development, we typically we think about the druggable proteome, right? So the druggable proteome is the number of active sites across all the proteins in our bodies that can be targeted and accessed by a small drug. Well, that number is 15%. 15% compared to 98% of the genome that has this unprecedented reach, not just in what it targets, but how they target those genes. And so this presents an incredible opportunity for us to learn and an incredible opportunity for us to develop novel therapeutics for diseases that have, been at an impasse for quite some time. Okay. Now, I'd like to talk about this paradigm, and we call it CREDO because it stands for cell-derived RNA from extracellular vesicles for bio-inspired drug development. I know it's a mouthful, but very simply speaking, again, we take EVs from therapeutic cells. We identify a single small RNA based on a assigned list of criteria. And then if there's a phenotype that exists, then we find out what the target cells are. We perform sequencing to figure out what the pathway is and how this RNA affects this pathway. And that constitutes a basic understanding. Once we've solidified A, how it works, and B, that it's, or rather, that it's therapeutic, and B, how it works, then we design a drug based on that. And for the next several slides, I'll give you just one example of an RNA that we did discover, how it works, and how we've made a drug out of it. Let me tell you very quickly about cardiosphere-derived cells. And cardiosphere-derived cells are these adult myocardial-derived stromal cells that are prepared simply by taking cardiac explants and then culturing them in an adherent culture. After a couple of weeks, EDCs or explant-derived cells begin to grow around this explant. And then we take those cells and then we culture them in semi-suspension, and they form these very beautiful three-dimensional cell spheres that we call cardiospheres. And then when we take those spheres and we culture them in adherent culture, they become this monolayer that we call CDCs, and that's the cell therapy that we've been working with actually long before I joined Eduardo's lab back in 2007. And these cells have advanced all the way to phase 3 clinical trials, and they passed phase 3 clinical trials for Duchenne muscular dystrophy. That's the HOPE-3 study that concluded fairly recently with success. And we should see the results soon published in a very good journal that escapes my memory at the moment. But anyway, when I was a graduate student, we were thinking about how CDCs function, and they function in a paracrine manner, but we didn't know what they were secreting. But long story short, CDCs function by secreting these extracellular vesicles, and we knew that the EVs were involved because we ran two important studies, one that demonstrated necessity. So we took CDCs, and in one group, we just gave them DMSO, that's the vehicle. And another group, we gave them this drug called GW4869, which inhibits ceramide synthesis and inhibits at least exosome secretion. And we gave those cells to mice that have a myocardial infarction or a heart attack. Now, normally CDCs, when they're given to animals that have a heart attack, their cardiac function improves, as you can see with the green line. But for some reason, the CDCs that couldn't secrete those EVs were no different than control. So that establishes necessity. Now we wondered if they were sufficient in place of the cells themselves. So we injected in the same model CDC EVs on their own and compared to fibroblast EVs. And as you can see, the fibroblast EVs were therapeutically inert, while the CDC EVs improved cardiac function 30 days post injury. Now, since then, we have validated the bioactivity of CDC EVs in several different models and in several different species, alongside our collaborators, including groups that we have no affiliation with, including this group from Japan. So time and again, and using different isolation methods, we established, excuse me, we established the bioactivity of these CDC EVs. So we're sure that they exert disease-modifying bioactivity. Now the question is how and why. So in mining the RNAs that are found in CDC EVs, we were very cognizant about not necessarily falling back on microRNAs. And the reason being is that many papers have shown that microRNAs can be a minority of CDC EVs. And certainly if you remember the pie chart I included earlier, they're not very ample. They're not very abundant. So the question was, well, what is abundant? And one fragment we found came from this yRNA4 gene, which we've never heard of. I mean, yRNA genes in general are understood to stabilize DNA during replication, and they're implicated in a few diseases by association only, and that's that. But because of its abundance, it really incentivized us to study what those are. And this one fragment stood apart from everything else. It's called EVYF1. And there was a smaller version of it called NT4. So we decided to study that particular fragment, NT4. And I didn't include this here, but it's cardiac protective in a rat model of myocardial infarction. So we knew it was abundant. We knew that it was therapeutic. It was time to make a drug out of it. So we took it through 3 stages of development. The first stage was to mutate the 5' residue to see if we can extend the therapeutic capability of this RNA drug. And then we added locked nucleic acids, alternating locked nucleic acids on both the 5' and the 3' end to enhance the stability of the RNA. And then the third, we tested methylating some of the residues, but that didn't make much of a difference. The assay that we use here to screen for the different variants are, excuse me, macrophages from rat bone marrow. And exposing them to the RNA would naturally raise IL-10 levels. So we use that as a surrogate marker for therapeutic capacity. In any case, if you look at the right bottom corner, you see the enhanced stability of this TY1, which is the bio-inspired drug of NT4 and EVYF1, which are the naturally occurring compounds. So we wanted to know how it worked. So we sequenced the transcriptome of these macrophages, and we found that one thing sort of popped out, which is this DNA damage response. Now we hadn't heard about, I mean, we were aware of DNA damage, but we weren't really aware that this was something that could be relevant to disease. And if you look at the heat map on the right, you find that the pathway that was turned on was the CGAS sting pathway. Again, we had never heard of it, but we decided to follow the data. And we went back to the rat MI model, and indeed, when you look at the animals that were given intravenous infusions of TY1, that TREX1 by protein was up, and then phospho-H2AX, which is a marker of DNA damage, was reduced in animals that were given TY1 compared to Vesicle and Scramble. Scramble, by the way, is the same nucleotide content as TY1, but jumbled. So you don't have that sequence specificity. And ideally, it shouldn't work if TY1 really is therapeutic. And so it reduces DNA damage and increases TREX1. Fine. But how? The next thing we decided to do is to see what RNA in the macrophage does this TY1 RNA bind. And we use 2 methods. One is called the bait capture method. And the beauty of this method is that all you need to do is to incubate the RNA with the biotinylated TY1 with macrophage lysate, and then just pull down the TY1 and see what it binds. So it's kind of a messy but very simple approach. The other one was a probe-based approach. So you can add TY1 to live macrophages, and then you can leach out the TY1 along with its RNA binding partner, and that gives you some measure of specificity. And then we did a Venn diagram and then looked at what was in the middle. And the gene that not only was in common between these 2 methods, but was also differentially regulated in the transcriptome of the data that I showed you earlier, was this gene again called TREX1. So when we did binding analysis between the 3' and the 5' UTR of TREX1, we found that TY1 binds the 5' UTR of TREX1, which if an RNA is increasing its expression, it usually binds at the 5'. If it's targeting it for degradation, usually the 3'. Now, how it enhances its expression remains equivocal, but we suspect that it intervenes during the translation step because we've done subsequent studies, and translation proteins do also tend to bind to TY1. The other question that we asked is, what were the target cells? And this is a very interesting question in EV therapy. The question is, when you inject very few EVs, is that enough to go around? And what we found at least with our study with EVs, is that most of the EVs are taken up by monocytes and macrophages, and that's what makes a difference. And so here we asked if macrophages were necessary for TY1 to function. The way that we did that is that there's this toxin called claudinate, which is this toxin in a liposome that you can inject in animals. And because phagocytes are they take up things a lot better than normal tissue. You can deplete an animal of its monocytes and macrophages, and to be fair, neutrophils as well and other phagocytes. But anyway, if you deplete rats of macrophages, and then you give them an MI, heart attack, and then you give them TY1, well, the animals that have intact macrophages, TY1 will reduce the infarct size after 48 hours. The animals that were given claudinate, i.e., they don't have functional macrophages, the effect of TY1 is attenuated. So that's, once again, that's necessity. Then specificity, we decided to adoptively transfer not TY1, but macrophages that were programmed by either vehicle, so just regular old macrophages, scramble, I mentioned earlier, TY1 and TREX1, which should function similarly because if TREX1 is a downstream effector, then upregulating TREX1 on its own should be therapeutic, and macrophages that were transfected with TY1 plus a knockdown of TREX1 to see if TY1 depends on TREX1 for its therapeutic capacity. And indeed, when you look at the right-hand figures, you find that only the macrophages that were programmed with TY1 or TREX1 were able to reduce the size of the infarct in these animals. So that establishes macrophages as critical and necessary mediators of TY1 therapy. But there was still some details that were left, which is, okay, imagine an animal has an injury in its myocardium and TY1 has just been uptaken by its macrophages. Okay, well then what? What happens then? And oddly enough, one of the early observations we made led us to believe that it goes back to EVs. So wasn't necessarily that the macrophages were homing to the area of the injury and doing the repair themselves. Certainly there was a little bit of that, but by and large, macrophages were secreting EVs that they themselves were now repairing the cardiac tissue. And the way that we found that out was, again, we went back and adoptively transferred macrophages that were either given vehicle or TY1 and given GW4869. So again, no EV secretion. And in these instances, we found that both the infarct size stayed the same and the cardiac troponin, which is a marker of cardiac damage in the blood, also remained the same. Interestingly, when you take TY1 and you give it to injured cardiomyocytes in culture, they don't respond. So cardiomyocytes will not stress cardiomyocytes will not show improved survival if you give them TY1 alone. However, if you give them EVs from macrophages that were programmed with TY1 or TREX1, then you begin to see a protective effect, which then led us to do the final study, which is to say, okay, forget the macrophages, forget TY1, let's give these animals EVs from macrophages that were given TY1 or TREX1. And once again, you see that the EVs themselves from the macrophages are the ones that do the repair. So to bring all of this together, when you have a trigger, an injury of some kind, it could be ischemia, it could be trauma, infection, whatever else, DNA damage goes up and cGAS sting also goes up. And that leads to inflammation and then downstream tissue fibrosis and remodeling and dysfunction and disease. However, when you add TY1, TREX1, which is this, forgive me if I forgot to tell you what TREX1 was, but it's a 3' exonuclease. It has two jobs. In the nucleus, it helps repair DNA damage. In the cytoplasm, it helps degrade DNA that is leaking from either the mitochondria or from the nucleus, and that helps suppress cGAS signaling, which downstream activates interferon and pro-inflammatory genes. But anyway, TREX1 goes up, DNA damage goes down, you get immunomodulation, and you get tissue repair. Now you can imagine all these sorts of injuries that involve DNA damage means that TY1 might be more broadly applicable than we've previously thought. And indeed, over the past 6-7 years, we found that not only is TY1 therapeutic in the context of myocardial infarction, but it's also therapeutic in the context of transplant rejection, Duchenne muscular dystrophy, hypertrophic cardiomyopathy, acute lung injury, and scleroderma, and heart failure with preserved ejection fraction. Now, we're moving towards the clinic now. We're trying to submit an investigational new drug application by the middle of this year, and we're actually going for the scleroderma indication because of the fact that it's a miserable disease and it's the deadliest autoimmune disease, and it's a good indication to go after. And the models that have been shown efficacy have included pig, rat, mouse, and also in culture. Of course, when you step away from EVs, you lose the advantage of delivery, right? So we have an RNA that's synthetically made. Now we have to find a way to formulate it to be able to deliver it in a number of ways. So classically, most of the studies were done using intravenous injection. So you formulate the RNA in a transfection reagent, and then you inject it retroorbitally, tail vein, whatever else. But lately, we've also developed a method of oral formulation. And it's very simple. We use the milk protein casein and the shellfish saccharide, chitosan. And the reason why these two ended up being very important is because casein seems to be very good at protecting the RNA as it goes through the digestive tract. And chitosan seems to optimize the TY1 for uptake by macrophages in the small intestine, namely the lamina propria and in the Paris patches. And when we did biodistribution studies, we find inexplicably that these that this TY1 ends up specifically in the PBMCs, but in nowhere else in the body, not even free TY1 in the blood, which we think this transfer happens in the Paris patches. And then we've also developed a way of injecting it subcutaneously. In this case, you don't really need a formulation. The locked nucleic acids are sufficient. Now, that is helpful because we've developed an entire pipeline of new drugs that can benefit from this delivery systems. So since TY1, we've discovered another RNA called YRX3, which we've made a drug out of called TY2. And we've discovered tRNA fragments, long non-coding RNA, and an unmapped sequence that we're currently studying. So there's a pipeline of RNAs, all from mining therapeutic EVs and learning how they work and learning where they can be applicable in terms of indication. So to recap the paradigm, we discover small RNAs from therapeutic EVs, we find out what the mechanism is, and then we develop a drug. Now, this is not only for the development of therapeutic candidates, it's also the identification of therapeutic targets. So imagine with me that you're someone studying pancreatic cancer. So let's say ductal adenocarcinoma, right? And you look at the EVs and you find an obscure RNA that is abundant, that is present in every sample that you find. That's not a therapeutic candidate, that's a target. Perhaps this small RNA is something that the tumor is addicted to. You find enough of these RNAs, you develop antisense against them, and you may have a chance of limiting the growth of this tumor. And so not only is this paradigm good for identifying therapeutic RNAs, but it's also good for finding pathogenic RNAs that you can then target and perhaps cut the tumor at the knee. So this is the versatility of this approach. So let me zoom out and discuss EV therapy in general. And that includes native EVs, that includes engineered EVs, that includes EVs as drug carriers, and so on. We can't ignore the reality that the field is ripe for change and progress. And the reason why I say that is on the economic side, you have venture capital investment that is very squeamish about investing in EV companies. And that's for a number of reasons. And that is, we're still somewhat in transition. Yes, 20 years have passed by, but we're still a very young discipline. When you look at, RNA vaccines began in the early 90s, and they've only saw fruition in the recent pandemic. And CRISPR is the same. Certainly, cell therapy is the same. And so we're still somewhat of a young field. Also, the current perception is that therapeutic EVs remain experimental because there have been studies that showed that uptake is very minimal, that microRNAs are too few to have an effect. But then again, this was in certain types of cells, and the discussion is still going on as to the generalizability of these findings. On the industry side, we're still wrangling with issues of understanding what the mechanism is. Excuse me. So is it the small EVs that are functional? Is it the large EVs that are functional? How much do we need to pare down to be able to have a clean therapeutic? So that creates a lot of bottlenecks. And it also creates a high cost of goods, especially if you're going to be manipulating these EVs for engineering. So there's definitely a technical need to make these therapeutics scalable. And then finally, again, an extension of this lack of understanding is the natural regulatory uncertainty. Discussions with the FDA tend to be very sort of, the FDA is like, well, teach us how your therapeutic works, and we'll work together to find out what is acceptable and what is not. That tends to be difficult if we don't know yet ourselves. And so I think all of these challenges boil down to the root questions. There's three of them, and that is, what is the role, for instance, of soluble proteins in EV signaling? And what role does the cargo play in the signaling effects? And what is the mechanism by which EVs exert their therapeutic capacity? And what I would like to offer is that there's no one answer that fits all. So instead of saying that, what is, we should be saying what are. Because again, some EVs work by engaging the receptors on the external, sort of on the plasma membrane. Others function by being internalized. Others function by, and the list goes on and on. So I think it's a lot more nuanced than we thought. And I think the current progress that we're making in understanding that nuance is going to be very, very key. So where do we go from here? Well, I believe that discussions must continue. Discussions in the circles of ICEV, AAEV, ASIC, all these societies that constantly engage the question of how can we make EV therapeutics scalable? How do we better understand the mechanism so we know what we're working with? And so on and so forth. So a science discipline is a lot like a relationship. The relationships don't die in yelling and screaming, but they die in silence. And so discussions must continue. And one of the most inspiring things that I've seen is the natural evolution of the MySev. So the first MySev was published in 2014. And when you read sort of the evolution of how we view EVs especially from 2018 to 2023, it's actually quite inspiring because there's a growing appreciation for the fact that different preparations are going to be relevant for different applications. And there's this growing value of the importance of diversity in how we not just understand EVs, but how we work with them for the different applications that they have. So I think that's pretty cool. And also the emergence, like as I mentioned before, of new societies with different ideas and different, perspectives. I think that adds more important discussions. And of course, today you'll be having discussions from two of the big leaders in EV translation, which is Susmita and Mario. I know both of them have learned a lot from them over the years, so this is ripe area for discussion. Furthermore, I think, I've already covered this, but aside from realizing the inherent diversity of EV sources and their preparations, we need to be we need to venture into uncharted territory. It always kind of breaks my heart every time I go to an EV conference, and 99.9% of the time, the cargo that's being investigated are microRNAs. There's more to the RNA cargome, if we want to call it that way, than microRNAs. And if we only look under the light post, we are unlikely to find anything useful. We also need to do a better job of incorporating all the new findings because papers are being published at a breakneck pace. And so when we go to conferences and we fail to corral all the findings, we're not equipped with all the information, and then the field tends to sort of stagnate. And then we also need to decouple, to some degree, our discussions of therapeutic translation from mechanistic objectives. Because even though these are two horses running the same race, they have different objectives, and thus they should be they should be free to ask their own questions separately. And then finally, we need creative ideas from bright scholars like you. This workshop is really to give you a lay of the land, and we make no pretensions about knowing all the answers. In fact, this you guys are what this whole workshop is all about, and we're hoping you can give us some answers yourself. And with that, I've a lot of people to thank. No man is an island. I see myself more as a peninsula, but anyway. I'd like to thank my mentor and collaborator, Eduardo Marban, who was my thesis mentor and now collaborator and the head of our institute. I'd like to thank every member of my lab. Alessandra just recently became a professor in her own right, and she earned a CERM grant as well as an R1. Hiro, Salwa, Josh, Deanna are all my amazing postdocs. Kara and Jess are my RAs, and Ann and Charlotte are my interns from UCLA. Also my collaborators, Eugenio and Russell, and the Smith-Hart Institute for their amazing support, Gina, Jane, and Cheryl. Of course, we're lucky to be here in California to have the wonderful support of the California public in the form of their taxpayer dollars in CERM, as well as the NIH. And of course, I have my warmest thanks to my two friends, Uta and Tanina, for their wonderful support and for always inviting me. I always have fun with them, and it's such a pleasure and privilege. Now, the important part is if you have any questions, or if you need any advice, or if you need a copy of this presentation or a PDF of any of my publications, this is my name, this is where I'm from, and this is my email. So feel free to email me anytime, and I really wish you the best of luck. This is a cool workshop. You'll learn a lot, and best of luck with your investigations and studies.
Thank you so much, Ahmed. This was wonderful as usual. And I have to say, I can speak for myself and for Uta. We always have fun and learn a lot from your presentations as well. So this is really nicely putting together. As Uta is walking in the room with the microphone and gathering questions, maybe we can, and we're running slightly behind the time, we can address those that were posted in the chat. So we have actually two questions from the same person, Jalil Zetouni. The first question was, in your experience, which EV-associated biomarkers are currently the most promising for near-term clinical translation, especially in non-invasive biofluids such as saliva and blood? I'm not sure we have the answer yet, like what is the best, but I will let you address the question.
Yeah, I guess I can benefit from some clarification. EV-associated biomarkers, you mean of the EVs themselves?
Yeah, like biomarkers inside. I guess the cargo, EV cargo that can be used as biomarker.
That's a great question. Well, in my humble opinion, as I've mentioned before, it's going to be different from one EV population and another. Really, I mean, especially in terms of cargo. If you're talking about surface, I mean, we've always used CD63, CD81, CD9, but when you're talking about cargo, it's so uniquely different that it's something that you would need to sort of figure that out for yourself because it's just going to be different from one population to another, even from one preparation to another. The one piece of advice I give the person asking the question is keep it as simple as possible, because remember, one of these days it'll be translated to the clinic. So in a clinical setting, you're very limited technically, so you won't have any flow to isolate EVs. The simpler the preparation, the better.
That's a good point. And then as a follow-up, given the role of macrophage reprogramming by EV-derived RNAs, do you see a similar mechanism being relevant in ischemic stroke
Absolutely.
and or
Abs-
post-stroke inflammation?
Yes, I do. Yes, indeed. Macrophages, and I guess in the context of stroke, microglia as well. They're not the first responders, but they're the ones that linger, and their dysregulation is what drives the disease pathology. So absolutely, yes, definitely.
Thank you so much. Uta, do we have any questions from the audience in person?
First of all, thank you so much for this inspiring talk. So, wow, I'm so close to this one.
Thank
So,
you.
is there someone for one question, some burning question here in the room? Otherwise, we'll move on. I'm sorry, I'm the reason why we are delayed today. So my introduction was too long. Any burning question right now? Otherwise, we'll go on to the next speakers. I think.
Yeah, and you guys have my email, so
yes,
feel free to
we'll
email
move
after.
on. Thank you so much.
Thank you.
so much.
Thanks,
and I know
everybody.
it's super early in your time zone, so I really appreciate it.
No, it was a pleasure.
Bye.
Bye, guys.
Thank you.
Bye.